Hello everyone, you are welcome to my YouTube channel. In continuation of the lecture series on basic microbiological tests, I'll be discussing characterization of Salmonella isolates today. Characterization of any microorganism involves steps in which the morphological characteristics, cultural characteristics, biochemical characteristics are determined. Others include serological characteristics and molecular characteristics. Salmonella are gram-negative motile rods that characteristically ferment glucose and mannose without producing gas, but do not ferment lactose or sucrose. So they are referred to as non-lactose fermenters. They are often pathogenic for humans or animals when ingested, particularly when raw or undercooked animal products are eaten. For example, some people engage in drinking raw egg yolk. Don't know what that is supposed to achieve, but some say that it will increase their intelligence quotient, but not knowing that they are at the risk of contracting salmonellosis. Salmonella is usually present in much lower numbers in food and water samples than in clinical specimens. It is to be noted that the surviving organisms are often injured or stressed, and so they have to be encouraged to grow using pre-enrichment broth and enrichment broth, as we'll see shortly. The primary detection of Salmonella isolates from food samples involve growing them in pre-enrichment broth, such as buffered peptone water, and then they're incubated. The tubes are incubated at 37 degrees Celsius for 24 hours. This is followed swiftly by the next step, which is enrichment in tetrathionate broth or selenite 16F broth. The culture is incubated at 42 to 43 degrees Celsius for 48 hours. Plating then occurs on selective media, such as bismuth sulfite agar and brilliant green agar. These plates are incubated at 37 degrees Celsius for 24 hours. Here is a summary of the flowchart of the methodology for detection of Salmonella isolates from food. The pre-enrichment, like I told you earlier, involves weighing 25 grams of the food sample at a ratio of 1 to 9. That means 25 grams of the food sample is mixed with the buffered peptone water, usually 225 mils, which gives 1 to 9 ratio. Now, a food homogenate is produced by blending this mixture, and then is incubated at 35 degrees Celsius for 24 hours. This is followed quickly by enrichment, in which the selenite tetrathionate broth is inoculated from the pre-enrichment broth and incubated at 43 degrees Celsius for 24 hours. Also, here, another tube can be inoculated, and the medium is Rappaport Bacillidis medium. The same conditions of incubation at 42 degrees Celsius for 24 hours. Plating is now done on bismuth sulfide agar, xylose lysine deoxychelate agar, and then incubated at 35 degrees Celsius for 24 hours. Further screening can be done using triple sugar ion agar, lysine decarboxylase agar slants. Confirmation is done by carrying out serological tests and other biochemical tests. That's for food samples. For water samples, the primary detection of Salmonella isolates can be done by using the membrane filtration assembly fitted with 0.45 micrometer and 250 milliliter from each water sample should be used per single filtration process. The filters are then inoculated on Salmonella chromogenic agar. The inoculated cultures should be incubated at 37 degrees Celsius for 24 hours. It is to be noted that Rappaport Bacillidis medium should not be used as the enrichment broth for the recovery of Salmonella typhi in water because it contains malachite green, which inhibits its growth. The primary detection of Salmonella isolates from clinical samples is done using selenite F broth, particularly stool specimens. The tubes are incubated at 37 degrees Celsius for 24 hours. It is important to state here that Salmonella organisms were first detected in clinical specimens, particularly in stool. That is why the F in selenite F broth stands for feces. The plating on McConkey agar, bismuth sulfide agar, xylose lysine deoxychelate agar, and Hection enteric agar is done. Salmonella form pink-red colonies on brilliant green agar. Hydrogen sulfide producing Salmonella form red colonies with black centers on XLD agar. Cultural characteristics of Salmonella isolates on some selective media. Looking at the Hection enteric agar, the colonies are blue-green to blue with or without black centers. On bismuth sulfide agar, typical Salmonella colonies may appear brown, gray, or black, sometimes with a metallic sheen. Some strains may produce green colonies with little or no darkening of the surrounding medium. You can see here, the plate 1 here shows the cultural characteristics of Salmonella isolates of Hection enteric medium. Looking at this, you see the evidence of hydrogen sulfide production in the center. Now let's quickly look at the biochemical identification of Salmonella isolate. The first biochemical test is the indole test. The procedure simply involves growing the pure culture of the test organism in a 5 ml culture medium containing the amino acid tryptophan. The culture media which can be used include peptone water, tryptone water, nutrient broth, trypticase soil broth. The inoculated culture medium is incubated at 37 degrees Celsius for 24 to 48 hours, to which 0.5 ml of COVAX reagent is added. The tube is shaken. A positive test is usually indicated by the appearance of a ring in the surface reagent layer or a red color above the broth within 1 minute. Now, what's the principle behind this test? If the test organism is able to produce an enzyme known as tryptophanase, these will now break down the amino acid tryptophan to liberate indole, pyruvic acid, and ammonia from tryptophan. The released indole reacts with the active para-dimethyl aminobenzaldehyde to form a red compound, which is seen in a positive test. For Salmonella, this is split 2. For Salmonella, it is indole negative. And for the other members of the Enterobacteriaceae, this is the reaction, a positive test. The alternate indole tests include: 1, the use of sulfide indole motility tests. That is 3 tests in 1. Motility indole ornithine tests, 3 tests in 1. Indole spot test. Now, the MR or methyl red test is also important. Salmonella isolate give positive MR test. The tube to be inoculated is a 5 ml methyl red Pascal broth and is incubated at 37 degrees Celsius for 48 to 72 hours. It is important that the culture must be incubated for at least 48 hours. After incubation, 1 ml of the MRVP broth is transferred into a small serological tube and 2 drops of methyl red indicator are added. A red coloration of the MRVP broth is a positive MR test, while a yellow color indicates a negative MR test. From this plate 3, it means that for a positive test, there is a red color in the tube developing after few minutes. And then you have a negative test, which is characteristics of Salmonella, no color change. Vospascal test is another important biochemical test for identifying Salmonella isolate. For half of the MRVP medium, 15 drops of 5% alpha naphthalate are added, followed by 5 drops of 40% potassium hydroxide. The tube should be shaken and the cap loosened and kept in a slanted position. The appearance of a red color within 1 hour indicates a Vospascal VP positive test. Salmonella isolates are known to give negative VP tests. Here it means that there will be no red color in the tube within 1 hour. Plate 5 shows urate tests. Salmonella isolates when inoculated, or the test organism when inoculated into urease broth. Urease agar slants can be used, but here we have the urease broth. If an organism is able to produce an enzyme urease, then it is able to degrade urea and releasing ammonia. Now, because of the presence of ammonia in the medium, the pH indicator, there will be a change in the color of the pH indicator, the phenol red. And so this is a positive test. But for Salmonella isolate, they give a negative urine test. Here is split 6 showing the motility test. The procedure involved inoculating the test organism with a fine stab along the soft or semi-solid medium. Now, if the organism is motile, as is indicated here, although this is the sulfide motility, Indo-motility medium. So evidence of motility is that there is no defined line of inoculation that can be seen at the initial introduction of the or inoculation of the organism into the tube of motility tube. So the medium is cloudy, and then there's evidence of even hydrogen sulfide production. Here is plate 7, which shows the beta-galactosidase test. Salmonella isolates are known to be negative. Here you have plate 8, which shows the lysine decarboxylase test. Now, Salmonella isolates are known to be positive. There is a deepening of the color of the medium because the lysine, the amino acid which is present in the medium, is decarboxylated by this enzyme lysine decarboxylate. And then there is a change in color from the yellow to purple. Reactions in triple sugar ion agar. Salmonella isolates are known to give an alkaline over acid reaction, that is K/A. And some serotypes are known to produce hydrogen sulfide, that's the darkening of the medium. Then for Salmonella, some serotypes are known to produce gas, although most of them don't produce gas from the glucose that is present in the triple sugar ion agar. Here is plate X showing the BBL enterotube identification for Salmonella enterica subspecies arizonae. Now, this is a tube containing 12 compartments. The first one shows evidence of glucose fermentation because of the yellow color. This is strictly for Salmonella enterica subspecies arizonae. Now, the next two to have the lysine decarboxylate medium and the ornithine decarboxylase medium. There's a change in color to the deep purple, meaning that Salmonella enterica subspecies arizonae is positive. Then you have the evidence of hydrogen sulfide production, see the blackening there, and all other tests, the sugar fermentation like dusitol and arabinose and sorbitol, excuse me. Serological identification of Salmonella isolate will involve the use of polyvalent somatic tests. And this briefly involves using Salmonella polyvalent somatic or antiserum to be added to where a loopful of the culture from a 24 to 48 hour triple sugar ion agar slant. This is mixed properly, emulsify the culture in saline solution and then you till the mixture back and forth for a minute, and then you observe for agglutination. Positive, it's Salmonella for evidence of polyvalent somatic antigen is positive if agglutination occurs in the text mixture, but no agglutination in the saline control. Same for polyvalent flagellar H test. Is a positive test for Salmonella if there's agglutination in the text medium and no agglutination in the control. The acceptable methods for the rapid identification of Salmonella isolate include API or Analytical Profile Index 20E, Enterobacteriaceae 2 system, Enterotube 2 identification system, the one that was explained earlier, and microID system. Immunological techniques and other protocols for the rapid identification of Salmonella isolate include immunodiffusion method, monoclonal enzyme immunoassay, both colorimetric and fluorogenic methods, polyclonal enzyme immunoassay. Other protocols include the proteomics and then the matrix-assisted LASA, desorption, ionization, time of flight mass spectrometry. Molecular methods for the rapid detection of Salmonella isolates include species-specific PCR, restriction, fragments, length polymorphism, nucleic acid hybridization method, nucleic acid sequencing. Here are some selected references and links for further reading. Thank you for listening.